வணக்கம் மாணவர்களே, இன்றைக்கி நம்ம மைக்ரோபயாலஜி சப்ஜெக்டில் மைக்ரோஸ்கோப்பி அப்படிங்கிற செகண்ட் லெசனில் இருக்கக்கூடிய கொஸ்டின் ஆன்சர்ஸ் தான் நம்ம பார்க்க போகிறோம். ஸோ இதில் புக் பேக் கொஸ்டின்ஸ், இன்சைட் கொஸ்டின்ஸ் எல்லாமே இதில் கவர் ஆகிடும். ஸோ நீங்கள் புக்கை வச்சுட்டு கரெக்டாக ஃபாலோ பண்ணிகிட்டே வாங்க. ஃபர்ஸ்ட் கொஸ்டின், வாட் இஸ் த ஃபேஸ் கான்ட்ராஸ்ட் மைக்ரோஸ்கோப்? வாட் இஸ் த ஸ்பெஷல் ஃபீச்சர் இன் இட்? This microscopy uses special optical components to exploit fine differences in the refractive indices of water and cytoplasmic components of living cells to produce contrast. It is a microscope which allows the observation of living cells. அதுதான் அதோட ஸ்பெஷல் ஃபீச்சர். ஸோ மற்ற மைக்ரோஸ்கோப்பியில் பார்த்தீங்கன்னா நம்ம பெர்மனன்ட் ஸ்லைட்ஸை வச்சு தான் நம்ம பார்ப்போம். நார்மலாக லைட் மைக்ரோஸ்கோப் ஒரு காம்பவுண்ட் மைக்ரோஸ்கோப்பில். பட் இந்த ஃபேஸ் கான்ட்ராஸ்ட் மைக்ரோஸ்கோப்பியில் மட்டும்தான் நம்ம லிவிங் செல்ஸை டைரெக்டாக நம்மளால் பார்க்க முடியும். ஸோ அதுதான் ஸ்பெஷல் ஃபீச்சர் ஆஃப் த ஃபேஸ் கான்ட்ராஸ்ட் மைக்ரோஸ்கோப்பி. நெக்ஸ்ட் கொஸ்டின், what is the principle of phase contrast microscopy? The phase contrast microscopy is based on the principle that small phases changes in the light rays induced by differences in the thickness and refractive index of the different parts of the object can be transformed into the differences in brightness or light intensity. The phase changes are not detectable to human eye, whereas the brightness or light intensity can be easily detected. ஸோ இதெல்லாம் உங்களுக்கு 2 மார்க் or 3 மார்க் கொஸ்டின்ஸில் கேட்கலாம். நேம் த ஆப்டிக்கல் காம்பனன்ட்ஸ் ஆஃப் ஃபேஸ் கான்ட்ராஸ்ட் மைக்ரோஸ்கோப்பி. ஸோ ஃபேஸ் கான்ட்ராஸ்ட் மைக்ரோஸ்கோப்பி is a similar microscope to the normal compound microscope. So it possesses a light source, condenser system, objective lens system and ocular lens system. So this can be asked in a 2 mark question. So next question, how does a phase contrast microscope differ from bright field microscope? A phase contrast microscope differs from a bright field microscope in having two different stages. One is a substage annular diaphragm and other one is phase plate. So these are the two components that is there in the phase contrast microscopy. So that is why it is different from the bright field microscope. And this can be asked in a 2 mark question. So அப்போ கேட்கும்போது இந்த ரெண்டு நேம்ஸ் மட்டும் எழுதினா போதும். இதே ஒரு 5 மார்க் or 3 மார்க்கில் கேட்கும்போது அதோட டிஸ்கிரிப்ஷனும் நீங்கள் எழுதணும். The annular aperture in the diaphragm is placed in the focal plane of the substage which control the illumination of the object. This located below the condenser of the microscope. This annular diaphragm helps to create a narrow hollow cone of light to illuminate the object. Next is phase plate. This plate is located at the back of the focal plane of the objective lens. This phase plate has 2 portions in which one is coated with light retarding material, that is like magnesium fluoride, and other portion devoid of light retarding material but can absorb the light. This plate helps to reduce the phase of the incident light. So this can be asked in a 2 mark or a 3 mark question. So next question, so working mechanism of the phase contrast microscopy. The unstained cells cannot create contrast under the normal microscope. When the light passes through an unstained cell, it encounters regions in the cell with different refractive indices and thickness. When light rays pass through an area of high refractive index, it deviates from its normal path and such light rays experience phase change or phase retardation. So retardation அப்படின்னா deviation. So the light வந்து deviate ஆகி போகிறது. The light rays pass through the area of less refractive index remain non-deviated. Shows the light path in the phase contrast microscope. The difference in the phases between the retarded light rays is about 1/4 of the original wavelength, and human eyes cannot detect these minute changes in the phase of light. The phase contrast microscope has special devices such as annular diaphragm and phase plate which convert these minute phase changes into brightness changes so that the contrast difference can be created in the final image. This contrast difference can be easily detected by human eyes. So in phase contrast microscope, to get contrast, the diffracted waves have to be separated from the direct waves. This separation is achieved by the substage annular diaphragm. The annular diaphragm illuminates the specimen with a hollow cone of light. Some rays pass through the inner region of the specimen and do not undergo any deviation, and they directly enter into the objective lens. The light rays passing through the denser region of the specimen get retarded and they run with a delayed phase than the non-deviated rays. Both the deviated and non-deviated light has to pass through the phase plate kept on the back focal plane of the objective to form the final image. The difference in the phase gives a contrast for clear visibility of the object. So, this will be asked in a 5-mark question, working principle of the phase contrast microscope. Next, what are the applications of the phase contrast microscope? So, these can be asked in a 2-mark or 3-mark questions. So, இதில் வந்து நீங்கள் applications நீங்கள் எல்லாமே படிச்சுக்கோங்க, அப்போ தான் வந்து 1 markல கேட்டாலும் உங்களுக்கு எழுத முடியும். So, phase contrast microscope enables the visualization of unstained living cells. It makes highly transparent objects more visible. It is used to examine various intracellular components of living cells relatively high resolution. It helps in studying cellular events such as cell division. It is used to visualize all types of cellular movements such as chromosomal and flagellar movements. Next question, this is a 2-mark question. Define fluorochrome. You know what is chrome? Chrome is color. Fluorescence means it gives the fluorescence, illumination. So, fluorescence microscope is a type of light microscope which instead of utilizing visible light to illuminate the specimen, uses a higher intensity light source that excites a fluorescence molecule called as fluoropore or also called as fluorochrome. So, next question, what is fluoropore? Fluorescence is a phenomenon that takes place when the substance absorbs light at a given wavelength and emits light at a higher wavelength. Thus, fluorescence microscopy combines the magnifying property of the light microscope with fluorescence technology. Next, this is a do you know question. So, what is Stokes shift? The British scientist Sir George G. Stokes first described the fluorescence in 1852. He observed that the fluoropore emitted red light when it was illuminated by ultraviolet excitation. Stokes noted that fluorescence emission always occurred at a longer wavelength than excitation light. This shift towards a longer wavelength is known as Stokes shift. So, this is a do you know box question. This is also a very important 2-mark question. Next question, what are the common fluoropores used? So, there are 3 types of common fluoropores used, that is diamidinophenyl lindole that emits blue color, FITC that emits green color, and Texas red that emits red color. So, the next question, what is the principle of the fluorescence microscopy? The principle behind the fluorescence microscopy is simple. The reflected light passes through an objective where it is focused on the fluorescent specimen. The emission from the specimen are in turn passed back up through the objective where magnification of the image occurs and now through the dichroic mirror. What are the components of fluorescence microscope? The main components of the fluorescent microscope resemble the traditional light microscope. So, அதே மாதிரி the normal compound microscope மாதிரியே தான் இந்த fluorescence microscope இருக்கும். But there are few special parts that can be modified in this. However, the 2 main differences are the type of light source used and the use of specialized filter elements. So, light source, what is the light source here? Fluorescence microscopy requires a very powerful light source such as xenon or mercury arc lamp. The light emitted from the mercury arc lamp will be 10 to 100 times brighter than most incandescent lamp and provides light in a wide range of wavelength from ultraviolet to the infrared. The lasers or high power LEDs were mostly used for the complex fluorescence microscopy techniques. Filter elements. A typical fluorescence microscope consists of 3 filters: excitation filter, emission filter and the dichroic beam splitter. The excitation filters is placed within the illumination path of the fluorescence microscope. Its purpose is to filter out all wavelength of the light source except for the excitation range of the fluorophore in the sample or specimen of interest. So, emission filter. The emission filter is placed within the imaging path of the fluorescence microscope. Its purpose is to filter out the entire excitation range and to transmit the emission range of the fluorescence, fluorophore in the specimen. The last one is dichroic filter or the beam splitter. The dichroic filter or the beam splitter is placed in between the excitation filter and emission filter at 45-degree angle. Its purpose is to reflect the excitation wavelength towards the fluorophore in the specimen and to transmit the emission wavelength towards the detector. So, இந்த questionலேருந்து பார்த்தீங்கன்னா நிறைய உங்களுக்கு 1 mark question இருக்குது. ஸோ இதில் வந்து என்ன விதமான லைட் சோர்ஸ் யூஸ் பண்ணுறாங்க, xenon and mercury arch lamp, this can be asked in the 1 mark question. அதே மாதிரி இதில் வந்து அந்த angle of the filter, that is 45-degree angle, excitation filter or the emission filter are placed at the 45-degree angle. So இதெல்லாம் வந்து 1 mark question கேட்கலாம். The next question. So this is also a do you know box question. So fluorescence is called cold light because it does not come from a hot source like an incandescent light bulb. So now fluorescence is also called as a cold light. Next, working mechanism of the fluorescence microscope. The specimen to be observed are stained or labeled with a fluorescent dye and then illuminated with high intensity ultraviolet light from mercury arch lamp. The light passes through the exciter filter that allows only blue light to pass through. Then the blue light reaches dichroic mirror and reflected downward the specimen. The specimen labeled with fluorescent dye absorbs the blue light and emits the green light. The emitted green light goes upward and passes through the dichroic mirror and reflects back blue light and allows only green light to pass the objective lens. Then it reaches barrier filter which allow only green light. The filtered fluorescent emission is sent to a detector where the image can be digitized. So this is the working mechanism of the fluorescent microscope. This is a 5 mark question. And next is the 2 types of fluorescence microscope. What are the types of fluorescence microscope? There are 2 types. One is diascopic fluorescence, another one is episcopic fluorescence. So these can be asked in a 2 mark question also. What are the types of the fluorescence microscope and who is the scientist who found it? K. Richard and O. Heimstad demonstrated the fluorescence microscope using autofluorescent specimens in 1911. This first type of fluorescence microscopy used transmitted light. The light from the illumination source first passes through an excitation filter and subsequently to the specimen through a dark field condenser. This eliminates most of the excitation light from the imaging side of the system. The next one is the episcopic fluorescence microscope. In episcopic fluorescence microscopy, the excitation light comes from above the specimen through the objective lens. So there is a difference between the diascopic and the episcopic. So what is the special feature in the episcopic? The light comes from the above the specimen through the objective lens. This is the most common form of fluorescence microscopy today. In this microscope, objective lens act as both condenser and objective. So quartz objective lens are required for deep ultraviolet excitation. So this is also a 1 mark question. What is the material of the lens that can be used for the ultraviolet excitation is the quartz. The next one is the application of fluorescence microscope. These can be asked in a 2 mark or a 3 mark question. So few 5 to 6 applications. You can write 3 points for a 3 mark question and 2 points for a 2 mark question. Fluorescence microscope has become one of the most powerful technique in biomedical research and clinical pathology. Fluorescence microscope allows the use of multicolor staining, labeling of structures within cells and the measurement of the physiological state of a cell. The fluorescence microscope help in observing texture and structure of the coal. to study the porosity of the ceramics using a fluorescent dye. To identify the Mycobacterium tuberculosis, this fluorescent microscope is also used. The next question. So, what is the difference between a light microscope and an electron microscope? So, you have 7 points. This can be asked in a 5-mark question. So, easily remembered. You have seen a light microscope. So, by seeing the light microscope, you can easily differentiate the electron microscope. You can easily remember the points of the light microscope and you try to remember from it. The first point, the light is the illuminating source here, and whereas in electron microscope the beam of electrons is the illuminating source. So, as the light is the illuminating source and here is the beam of electrons. Specimen preparation takes usually few minutes to hours. Live or dead specimen can be seen. In case of electron microscope, specimen preparation usually takes a few days. Only dead or dried specimen are seen. So, நம்ம நார்மலாக வாட்டரில் இருக்கக்கூடிய மில்கில் இருக்கக்கூடிய ஆர்கானிசமை கூட நம்ம லைட் மைக்ரோஸ்கோப்பில் பார்க்க முடியும். பட் இன் கேஸ் ஆஃப் எலக்ட்ரான் மைக்ரோஸ்கோப் யூ கேன் நாட் see. And condenser, objective and eyepiece lenses are made up of glasses, and in case of electron microscope all the lenses are electromagnetic. Next is specimen is stained by colored dyes, and here specimen is coated with heavy metals in order to reflect electrons. It has low resolving power. Its magnification is of 500x to 1500x. It has high resolving power, about 250 times higher than the light microscope. It has a magnification more than 1 lakh x. Vacuum is not required. Vacuum is essential for its operation. Image is seen by eyes through ocular lens. Image is produced on fluorescent screen or photographic plate. In electron microscope, we can see the cellular components like nucleus, plasma membrane and mitochondria. It is achieved by electron microscope, which have a greater resolving power than the light microscopes and can obtain higher magnification. Next question. What are the types of electron microscope? There are 3 types of electron microscope. One is transmission electron microscope, scanning electron microscope and scanning transmission electron microscope. Next question, the principle. What is the principle of electron microscope? The fundamental principle of electron microscope is similar to the light microscope. In electron microscope, a high velocity beam of electrons is used instead of photons. In the electron gun, electrons are emitted from the surface of the cathode and accelerated towards the anode by high voltage to form a high energy electron beam. All lenses in the electron microscope are electromagnetic. Charged electrons interact with the magnetic field and magnetic force focuses an electron beam. The condenser lens system controls the beam diameter and convergence angles of the beam incident on a specimen. The image is formed either by using transmitted beam or by using the diffracted beam. The image is magnified and focused onto an imaging device such as fluorescent screen on a layer of photographic film or to be detected by a sensor. So, next question, write a note on preparation of sample for electron microscope. So, how do you prepare a sample for an electron microscope? So, preparation of specimen is the most complicated and skillful step in electron microscope. The materials to be studied under the electron microscope must be well preserved, fixed, completely dehydrated, ultra thin and impregnated with the heavy metals that sharpen the difference among the various organelles. The material is preserved by fixation with glutaraldehyde and then with osmium tetroxide. So, this can be asked in a 1-mark question. What are the materials used to fix the specimen for electron microscope? The fixed material is dehydrated and then embedded in plastic and sectioned with the help of diamond or glass razor of ultra microtome. Okay, the next question, write a note on electron sample interaction. So, how does the electrons and the sample in the slide will interact with each other in the electron microscope? The interactions of the electron beam with the sample results in different types of electrons. So, different types of electrons are produced. One is elastic scattered electrons, inelastic scattered electrons, secondary electrons and back scattered electrons. Depending upon the kind of radiation or emitted electrons which are used for the detection, different properties of the specimen such as topography, elemental composition can be concluded. In transmission electron microscope, a beam of electrons is transmitted through an ultra thin specimen interacting with the specimen as it passes through. An image is formed from the interaction of the transmitted unscattered electrons through the specimen. The secondary electrons are mainly used in scanning electron microscopy for imaging the surface topography of biological specimens. The interaction of the electron beam with the samples result in secondary electrons and backscattered electrons and that are detected by standard scanning electron microscope equipment. The next question, write a note on working principle of the instrumentation of transmission electron microscopy. A transmission electron microscope has the following components: electron gun, condenser lens, specimen stage, objective lens and projector lens. Screen or photographic film or camera will be there to see the image. Electron gun consists of tungsten filament or a cathode that emits electrons on receiving high voltage electric current, and high voltage between the electron source and an anode plate is applied, leading to an electrostatic field through which the electrons are accelerated. The emitted electrons travel through the vacuum in the microscope column. The vacuum is essential to prevent the strong scattering of the electrons by gases. The electromagnetic condenser lens focuses the electrons into a very thin beam. The electron beam then travels through the specimen and then through the electromagnetic objective lens. In a transmission electron microscope, the sample is located in the middle of the column. At the bottom of the microscope, unscattered electrons hit the fluorescent screen, giving image of the specimen with its different parts, displayed in varied darkness according to their density. The image can be studied directly or photographed or digitally recorded. So, this is the principle and instrumentation of transmission electron microscopy, and this can be asked in a 5-mark question. Next, what are the information that can be obtained using transmission electron microscopy? So, in which fields we can use this transmission electron microscopy? The information that can be obtained using transmission electron microscopy are: one is topography, surface textures or surface features, morphology, shape and size of the particles, crystallographic arrangement of the atoms, and composition, elements and their relative amounts. Next, there is a difference between scanning electron microscopy and transmission electron microscopy. So, various properties are listed out and the difference are being given. So, first property is types of electron. It is based on scattered electrons that are emitted from a source of a specimen. It is based on the transmitted electrons. Second is the sample. The sample can be of any thickness and is coated with a thin layer of heavy metal such as gold or palladium and mounted on an aluminum slab. Laborious sample preparation is required. The sample has to be cut into thin section so as to allow electrons to pass through it and supported to the transmission electron microscopy grids. Resolution. The resolution is up to 20 nanometers. Transmission electron microscope has such higher resolution than scanning electron microscope. It can resolve objects as close as 1 nanometer. Magnification. The magnifying power of scanning electron microscope is up to 1 lakh x. The magnifying power of transmission electron microscopy is up to 50 lakh x. Next, the image formation. Scanning electron microscope provides a 3-dimensional image. Secondary or backscattered electrons are captured, detected and displayed on computer screen. Transmission electron microscopy provides a 2-dimensional image and transmitted electrons hit a fluorescent screen, giving rise to a shadow image. The image can be studied directly by the operator or photographed with a camera. The next point is application. So, scanning electron microscopy is used to study the topography and the atomic composition of the specimen. Transmission electron microscopy is used to study the interior of the cells, the structure of the protein molecule, the organization of the molecules in viruses and cytoskeletal filaments, and arranged of protein molecules in cell membranes. Next, working principle of the scanning electron microscopy. So, it is used to study the three-dimensional image of the surface of the cells, tissues or particles. Scanning electron microscope allows viewing of the surfaces of specimen without sectioning. The specimen is first fixed in a liquid propane at 180 degrees Celsius and then dehydrated in alcohol at 70 degrees Celsius. This can be asked in a 1-mark question, that is in which liquid the specimen is fixed, that is in liquid propane at 180 degrees Celsius. The dried specimen is then coated with a thin film of heavy metal such as platinum or gold. By evaporation in the vacuum provides a reflecting surface of the electrons. In scanning electron microscopy, samples are positioned at the bottom of the electron column and the scattered electrons are captured by the electron detectors. In the scanning electron microscope, there are several electromagnetic lenses including condenser lenses and one objective lens. Electromagnetic lenses are for electron probe formation and two condenser lenses reduce the crossover diameter of the electron beam. The objective lens further reduces the cross section of the electron beam and focuses the electron beam as probe on the specimen surface. The objective lens thus functions like a condenser. This is the contrast to transmission electron microscope where objective lens does the magnification. Scanning electron microscope are equipped with an energy dispersive spectrometer detection system which is able to detect and display most of the X-ray spectrum. The detector normally consists of semiconducting silicon or germanium. Next, write a note on scanning transmission electron microscope. The scanning transmission electron microscope combines the principles of transmission electron microscopy and scanning electron microscopy and can be formed on either type of the instrument. It is one of its principal advantages over transmission electron microscopy. This microscope requires very thin samples and looks primary at the beam electrons transmitted by the sample. One of its principal advantages over the transmission electron microscopy is in enabling the use of signals that cannot be spatially correlated in transmission electron microscope, including the secondary electrons, scattered beam electrons, characteristic X-rays and electron energy loss. It's a 2-mark question. What are the functions of barrier filter? The light is filtered by the barrier filter which selects for the emission wavelength and filters out contaminating the light from the arc lamp or the other sources that are reflected off from the microscope components. Finally, the filtered fluorescent emission is sent to a detector where the image can be digitized. So, this is the function of the barrier filter. It filters the light from the arc lamp. Second lesson of microbiology la microscopy அப்படிங்கற அந்த சாப்டர்ல இருக்கக்கூடிய 2 mark, 3 mark, 5 marks and 1 marks inside the book அதெல்லாமே வந்து இப்போ டிஸ்கஸ் பண்ணிட்டோம். இதுக்கு அடுத்த லெசன்ல நம்ம மீண்டும் சந்திப்போம். நன்றி, வணக்கம்.